Hi guys, so my name is Sarai. Um, I am a famous online student. Well, not famous, but I am the online student. The reason that I am not attending the classes with you guys is because I reside in Hawaii, so I really can't go to classes every day. It would be pretty expensive for me. But nevertheless, I am going to be talking about gene editing today. So we're gonna go ahead and talk about the definition of gene editing. We're gonna talk about some techniques, the discovery of CRISPR, which is what we're gonna be focusing on today. Um, we're going to talk about its components and how it works in bacteria, which is where it was discovered. And then specifically, I'm going to be talking about CRISPR-Cas9 and how it works. And then we're also going to be talking about the advantages and disadvantages of using CRISPR-Cas9. After that, we will go ahead and talk a little bit about a new gene editing technique that just came out last week called prime editing. Uh, and then we're going to go ahead and talk about the applications and the ethical concerns of um, CRISPR-Cas9. So genome editing is a technique that is used to modify DNA within a cell or organism. It either adds, removes, or alters DNA at a specific sequence. The first thing that we're going to be talking about are transposons. So transposons are segments of a chromosome that can be transposed to a new location. Um, and the way that this is done is pretty much by a cut and paste mechanism. Right now, transposons are used in genetic editing by insertional mutagenesis, which pretty much just uses the transposon as a vector to insert or remove DNA sequences at a desired place. Um, zinc finger nucleases are engineered DNA binding proteins that can bind to a variety of DNA sequences. They do create a double-strand break that is introduced at a specific site, and they are also composed of two things: the DNA binding domain and then the DNA cleaving domain. So if we look at letter A, here we can find a zinc finger nuclease repeats, and usually the binding domain, which is this, can be composed of 3 to 6 finger nucleases repeats, zinc finger repeats, and each repeat can bind to 3 base pairs. Then the second part is the DNA binding nuclease domain, which is the FOLK1 endonuclease. This nuclease, in order for cleavage to occur, has to dimerize. So these two have to dimerize, and then cleavage would occur. If they don't dimerize, then no cleavage is gonna happen. So the next one is talins, or talents. Uh, so these are transcription activator-like effector nucleases, and they also create a double-strand break that is introduced at a specific location in the genome. They are also composed of two factors, one of them being the tall effector DNA domain, and then the other one, the FOLK1 DNA cleavage domain. So the tall effectors is composed of two parts. One is the N-terminal region, which is responsible for the DNA binding, for the talent binding to the DNA. And then the second one is the central repeat domain. So the central repeat domain is composed of a number of modules, and each module has 34 amino acids. So amino acids that are located at amino acids 12 and 13 are responsible for the one nucleotide binding to the DNA. And then this is called RBDs, which stands for repeated variable diet residue. So as we can see, uh, each domain, like I said, only binds to one, which module only binds to one nucleotide, as opposed to in the zinc finger race, you can see that each zinc finger binds to 3 base pairs. So this is a difference between the talons and the zinc finger nucleases, but they both use the FOLK1 DNA cleavage domain to cut the DNA. So just like before for zinc finger nucleases, if these two don't dimerize, then there's no cleavage is gonna happen. So we're gonna go ahead and talk about CRISPR now. So what CRISPR is, or what it stands for, is clustered regulatory interspace short palindromic repeats. So what this is pretty much is just a mechanism that is used by bacteria to fight against phage infection. So the reason why CRISPR is showing to be more used right now is because it is very cheap, it's more efficient, it's more customizable, and it does not need to be paired with different artificial enzymes because it secretes its own, which is going to be Cas9 that we're going to be talking about later on. So we're going to go ahead and talk about the discovery timeline of CRISPR. In 1987, some Japanese scientists saw that there was a pattern of DNA in E. coli. And this pattern consisted of 5 short repeating segments of DNA sequence that were separated by non-repeating DNA sequences that are called spacers. So then in 1990, they discovered that many of the microbes had the same pattern. And then in 2002, some Dutch scientists finally gave this repeating sequence its name of CRISPR, and they also noted that these repeating sequences came with some Cas genes. And then in 2005, scientists discovered that the spacers sequence was very similar to viral DNA sequences. So in 2007, an experiment was done where some microbes were infected with viral DNA, or by a virus. They were infected by a virus, and they did notice that a lot of bacteria died, but they also noticed that some bacteria also survived. So further research showed that the bacteria who had survived had incorporated the viral DNA into its own genome, and that's how it helped it survive. So this was like a very important discovery for genome editing. So then we move forward to 2008, which was when the CRISPR-Cas9 mechanism was explained for the first time. And then in 2012, a scientist named Jennifer Doudna, she published her paper that demonstrated how CRISPR-Cas9 can be used for genome editing. And then in 2015, there was a huge ethical concern because it was reported that there were some genes being edited in human embryos. So this caused a huge problem in the science world. But then we go ahead and move back to now, and a new DNA technology has been discovered. It doesn't have to do with CRISPR, but it's still a genetic engineering technology that has been discovered, which is called prime editing. So we are going to go ahead and talk about it briefly in this presentation, but then we're also going to go ahead and talk about it more on Tuesday with our research paper. So the components of a CRISPR system, it has three main parts: the CRISPR array, the Cas operon, and the tracRNA. So the CRISPR array, which is right here, is composed of two things: the spacers and then the repeats. And the Cas operon would be here, and then the Cas operon has many Cas genes. And then we have the tracRNA, which serves as a function of binding scaffold for Cas9. So it helps Cas9 bind to the desired place in the genome. So the way that this works is that if we have a bacteria that has infected, if we have a bacteriophage or a virus that has infected the bacteria, usually what would happen is that the virus would replicate in the bacteria, and then eventually the bacteria cell would die. But with the bacteria who have this CRISPR system, what happens is when the bacteria injects its genome into, when the virus injects its genome into the bacteria, the system sends the Cas1 and Cas2 to go ahead and cut this DNA sequence from the virus, and then it incorporated into the CRISPR array. So once the sequence or the spacer is incorporated into the CRISPR array, it's then transcribed with all of the other spacers, and it becomes a mature cRNA. So when this virus comes again and injects its genome into the bacteria can now recognize what it's being inserted, and it go ahead and activates Cas9, which will come and cut the sequence, therefore killing the virus, not preventing it or not letting it replicate. So the Cas9 recognizes that this spacer with the PAM region, because the PAM region is very important for Cas9 to recognize where it has to cut. So this is how it works. This is how it was discovered in the bacteria cells. So like I mentioned before, there are many Cas genes. There are 93 Cas genes, and these are grouped into 35 families based on the encoded proteins that they make. 11 of the 35 families form the Cas cores, which includes Cas1 to Cas9. And then the CRISPR-Cas system falls into two classes. So the Class 1 system uses a complex of multiple Cas proteins to degrade foreign nucleic acid. But the Class 2 system uses just one single large Cas protein. So for our purposes, for whatever we're going to be talking about, we're going to be talking about CRISPR-Cas9, which falls under Class 2 subtype 2. So there, like they said, there are many different Cas genes, and this is just like a table that can show you the different amounts of Cas and the nucleases that are there. I mean, there are a lot that you know the function, but there are some that you still don't know the function of. But there were just too many Cas genes to talk about them all. So this is why we're only going to be talking about CRISPR-Cas9, which is here. So CRISPR-Cas9 uses the Cas9 enzyme, which is just pretty much the molecular scissors, and it is composed of two domains. The H and H nuclease domain and the RUBC nuclease domain. And then also another part of the CRISPR-Cas9 complex is the guided RNA, which pretty much just guides the Cas9 to the target site. So the H and H nuclease domain cleaves the DNA strand complementary to the gRNA. So this is our target strand. And then the RUBC nuclease domain just cleaves the strand that's opposite to the complementary strand. So there are also different Cas9 systems. For example, we have CRISPR-I, which is a form of the CRISPR system in which the Cas9 enzyme is inactivated, and this inactivation blocks transcription elongation or knockdown transcripts. We also have CRISPR-A, which activates the dCas9, and therefore transcription is increased. And then we also have the CRISPR-Cas9 nickase, which is very similar to the CRISPR-Cas9, which is the wild type, but it differs that it only cuts a single strand, so it makes a single DNA strand break. So the way that this works is that scientists first have to engineer the guided RNA strand, but when they are engineering this strand, they have to take into consideration the PAM site. Like I mentioned before, if the Cas9 does not recognize the PAM site, it will not bind, or will not then check to see if its RNA matches the DNA that it's trying to cut. So it is very important for the PAM site to be incorporated into this gRNA. So after the Cas9 finds this PAM site, it checks to see if its RNA matches the DNA found in the cell, and once it does that, it creates an RNA and DNA hybrid. So then this triggers the Cas9 to be activated, and then therefore cleaves at the desired sequences or at the desired place. So after this cleavage, the cell can either repair itself through non-homologous end joining or through homologous direct repair, which is when we grab a donor DNA sequence and incorporate it into the cell. So the advantages versus the disadvantages, we have many. And the advantages, it is more simple to use CRISPR-Cas9 because of the target design, which you're doing once you know where you want to cut, you can easily engineer the guided RNA. You can also do multiple target sites in one time, and then editing different human cells is possible with CRISPR-Cas9 because it works in many organisms and many cells. So that's one of the big ones. Some disadvantages that we have is there is a limited number of PAM sequences that are known, and like I mentioned before, the PAM sequence is very important for cleavage to occur. There's also some off-site effects that happen with CRISPR-Cas9, which the off-site effects can trigger mutations at undesired places. So because our DNA sequences are similar in other parts of the genome, the guided RNA can end up binding to a place you didn't desire to bind to, and therefore the Cas9 nuclease will cut at undesired places, which can cause mutations and things that we don't know how it might affect the cell. So the off-site effects are a big thing, and this is where prime editing comes in, which we'll discuss in a bit. Another disadvantage is that you're editing certain cells, but not all. So you're making a change in one cell, but there are also cells that are gonna continue to have that same problem. And then we of course have a lot of ethical issues that come with genome editing and using CRISPR-Cas9. So prime editing is the new, most fresh genome editing technology that just came out on the 21st of this month. So this is very important because it lowers the off-target effects that can be caused because of CRISPR-Cas9. So the way that this works, it's kind of like a search and replace mechanism. You do not create a double-strand break, and you do not need any donor DNA templates to fix this break. The complex is composed of an impaired Cas9. So what they did is that they modify the Cas9 to only cut at one strand. So it just cuts one strand of DNA, and then they also attached it to a reverse transcriptase, which is guided by the prime editing guided RNA, which we're going to be talking about later. So, as I mentioned before, the prime editor complex includes the Cas9, and then, I should say the edited Cas9 and the reverse transcriptase. With this, we also have the engineered PEG RNA, which directs the Cas9 to nick a DNA strand at a desired place. So the PEG RNA is composed of two things, the binding region and the editing sequence. So the binding regions is the section that binds to the nicked DNA, and then we also have the edited sequence, which is the section that contains the RNA letters that encode the desired edit. So the way that this happens is after the Cas9 nicks the one DNA strand, RNA transcriptase goes ahead and reads the RNA and attaches the corresponding DNA letters to the end of the nicked DNA. After that is done, and then the nucleus from the cell naturally excises the old segment, and then it seals the new letters into the genome. So this creates a problem because we have a mismatch. So the way that they correct this problem is by sending a different guided RNA which directs the complex to cut the other strand of DNA. So then after this other strand is cut, the cell pretty much goes ahead and corrects the strand by using the edited strand that was made. And pretty much prime editing technology seems to be safer to be used in a clinical in clinical technologies or in the clinic for therapies because of its precise changes and because it lowers that off-target effects that can be caused when CRISPR-Cas9 is used. So we're gonna go ahead and go back to the applications of CRISPR-Cas9. Uh, it has been used in many different places, such as bacteria, plants, insects, fish, reptiles, birds, and mammals. Uh, uh, CRISPR-Cas9 can modify somatic genes, which is pretty much what we're doing right now, and germline cells, which is an ethical issue that we're going to be discussing later on. There are many applications where CRISPR-Cas9 has been used, but we're going to be talking about agriculture and gene therapy. So pretty much this is just like an overall overview of the method that we will use when we use CRISPR-Cas9 system. We would first select the genome target, and then we would go ahead and design our guided RNA. After that, we would assemble it with the Cas9. We would assemble the Cas9 with the guided RNA and then deliver it to the plants. After it's delivered to the plants, we would regenerate and screen the transgenic plant for gene editing. So in agriculture, CRISPR-Cas9 is pretty much used for resistance, to create resistance for viruses, for diseases, for physical and chemical, such as like herbicides and stuff like that. They also use it for crop production enhancement. So we can see how CRISPR-Cas9 is really benefiting the agriculture field. The next one is using CRISPR-Cas9 in gene therapy. They have used it for sickle cell anemia because sickle cell anemia is a disease where we have a single nucleotide mutation in the HBV gene. They have used CRISPR-Cas9 to change this mutation or to alter this gene. They have also used it in HIV, and well, the scientists believe that if they edit the LTR promoter region, they can deactivate viral transcription. So what they did was that in an experiment with mice, they went ahead and removed the LTR, and so it's like a segment between the LTR and the GAG regions were successfully removed in HBI transgenic mice using CRISPR-Cas9. And when this section was removed, they found that there was a drastic reduction of HIV RNA. So they did find it very efficient. They have also used CRISPR-Cas9 in cancer. They have used it as a diagnostic marker to inhibit cell proliferation and then to generate cancer models. They have also used genome screening, and they have used CRISPR system with that. And the way that this is done is that, for example, in the cancer patients who exhibit certain mutations or alter gene expressions, they respond to that target drug treatment. But we also can't forget that cells can eventually become resistant to this treatment. So in order to fix this problem, what they do is that they use the CRISPR system to reveal the gene expression changes that have occurred after the treatment. And this will help them pinpoint the genes that are associated with resistance of the target drugs, and then they can therefore identify new biomarkers for precision therapy. So this is just a picture of the percentages of cancers that are being studied with CRISPR-Cas9. Pretty much a lot of the research with CRISPR-Cas9 is done in cancer. Uh, this is just like an idea that we get for CRISPR-Cas9 is being used. So now we're gonna go ahead and move to the ethical concerns. One of the big ethical concerns is safety. Because in CRISPR-Cas9, we have the off-target mutations. We don't know what mutations can occur because of the cleavage that we are doing. So when we are thinking of using this method for therapies, we have to outweigh the benefits over the risks. So the benefits have to be much greater than the risks. Another ethical concern is ecological disequilibrium. So because we are editing certain organisms, well, we are kind of worried that these organisms that are being edited can potentially then transfer their modified sequence to other species and therefore create an equilibrium in our ecosystem, because we're using some organisms over the others. One of the big ethical concerns that we have right now is editing human germline cells. So pretty much right now we are using somatic cells for therapeutic interventions, and the reason that we are not going into the germline editing is because we do not know what mutations and side effects can occur to the future generations. Not only that, but there is a problem with the informed consent, because you're editing germline cells, which are like eggs, the sperm, the fertilized eggs. You can't ask them for their consent. Obviously, it's not like you're gonna be like, well, you sperm, do you give me your permission to edit your genome, even though you're not capable of telling me you're giving me your permission and knowing what I'm going to be doing to you. So I mean, it is an ethical concern that arises when there is no really informed consent that is given. So because the CRISPR-Cas9 technology and genome editing technology is still very new, and we don't know much about its potential effects, the scientists are calling for a moratorium on applying CRISPR-Cas9 to the human germlines because of these problems. There's still things that we don't know, so they're calling on that moratorium. But in 2018, there's a Chinese scientist whose name is He Jiankui, who came out in 2018 in November saying that he had successfully edited the genome of two twin embryos, which was a bomb to the science community for many reasons. For ethical reasons, many people said that he crossed the line. You can't do that. So apparently the way that he did this was that he was working in a clinical research with couples who were trying to conceive but couldn't. So what he would offer was that he would fertilize an egg, obviously insert into a woman, but he would genetically alter one gene in that egg. So in the consent forms he wrote, and I quote, it would give the ability of the embryo to be resistant to AIDS. So what he did, oh wait, one of the requirements that he had was that the father had to be infected and the mother, she didn't have to be infected for the participants to be able to participate in this experiment. So what he actually did was that he went ahead and edited the CCR5 gene, which is a gene that encodes for a protein that HIV uses to come inside of the cell. But the problem is, other than the ethical concerns, that the HIV can use different proteins to come inside of the cell. So technically, the girls were not going to be resistant to AIDS or HIV. So a lot of the people were concerned because of the health of the babies, of the girls. They didn't know what potential effects or mutations can occur because of these editing that he had done to them. So pretty much his lab was canceled, he was closed, and everything was closed. The institution that he was working for did not know what he was doing, and they say that he was conducting the lab at another place, so they were not aware of this experiment. But yeah, this is one of the main reasons of why they went ahead and called for that pause in adding or using CRISPR-Cas9 in germline cells. So another ethical concern is the genome editing for enhancement. So not necessarily for health issues, but just for like traits that you would want to see in a person. Is it ethical to do that? Which then calls into the problem of eugenics, favoring one trait or one type over the other. So that is an ethical concern. And then we also have religion, which is another concern that there is. Are we, Christianity believes that God is the one who created humans, so editing humans would make us like gods, and we're intervening in God's creation. So it does create a concern for that. Not only that, but if the editing leads to a death, it's also a problem with religion. So there are many ethical concerns regarding genome editing and using CRISPR-Cas9 that arise. But as we talked before, there are also many advantages of it, especially when it comes to using CRISPR-Cas9 to treat diseases or to prevent diseases. For example, if when the two parents are homozygous and then their kids are all going to be affected, and then the thought of using CRISPR-Cas9 to prevent this is there, because it can potentially help these people. So this is pretty much it for this presentation. Like I mentioned before, we're gonna go ahead and talk more about prime editing on Tuesday's research paper. And well, I'll see you guys on Tuesday.